Thank you very much for inviting me, Heather. Here is something completely different, just to give you an idea of what can happen in genetic conditions in terms of other sorts of research. So the condition that I have been researching is called SYNGAP1 related intellectual disability. I'm just going to call it SYNGAP1 so it's not so much of a mouthful. It's another condition that is caused by changes in a single gene. It's not changing. There we are. And this is the SYNGAP gene. So it's a gene that codes for a protein called SYNGAP1, and that is found in the brain at synapses, which are the junctions between different neurons, different brain cells. It's a protein that interacts with many, many other proteins, and it has a role in regulating the transmission of signals from one brain cell to the next. In people who are affected by changes in the SYNGAP gene and therefore have SYNGAP1 related intellectual disability, they often have intellectual disability that is moderate to severe, although I just have a sense that we're now starting to identify people who have a bit more of a milder phenotype, so milder presentation, which is probably because people are being tested in terms of their genetics for a wider range of presentations. At the moment, at the end of 2023, the official census recorded by the SYNGAP Research Fund, which is a large family support network and research funder for SYNGAP, their census showed that there were 1,339 people worldwide who had signed up to say, I have a diagnosis. We know there will be many more people than that because there'll be people who haven't opted into that, but also, as has already been said, people who are older, so perhaps older children, but certainly adults are less likely to have been offered genetic testing in the past and therefore may well have SYNGAP without knowing it. So my research is centered around sleep. This was mainly because looking at the research papers for SYNGAP, you would have a list of symptoms that people were suffering from, and sleep problems were highly prevalent, but nobody had sort of systematically looked at that yet. So before we actually look at the research, we need to define what we're talking about. So we all know what sleep is, but actually when I sat down to write the talk, I realized I couldn't give you a definite sort of description of what it is. So the Cambridge Dictionary tells us that sleep is the resting state in which the body is not active and the mind is unconscious. Now, obviously, we spend a significant amount of our lives in sleep, some more than others. So it must be important. What does it do? Well, we know that if sleep is disrupted or the sleep-wake cycle, the circadian rhythm is disrupted, it can have all sorts of effects, and we know what it feels like, all of us, but specifically things like concentration, mood, ability to learn, general health, and also it can impair your immune function and make you more susceptible to things like infections. So I wanted to look at whether you could find biomarkers, so sort of specific patterns in SYNGAP1 sleep that you couldn't identify in people without that condition. And I will come back to the fact that I was thinking about this cross species a bit later on, but it's because I have collaborated with Dr. Alfredo Gonzalez Sousa, who is also at the University of Edinburgh, who was looking at SYNGAP1 rats and how well they sleep. So what did we do? We had a variety of different things. So we sent out questionnaires to participants across the UK. They included medical information, medication, development, social information, so the Children's Sleep Habits questionnaire, and a questionnaire about quality of life. We asked participants to wear a actigraphy watch for 7 days. So that's a bit like a Fitbit. You wear it literally like a watch. It is just a Velcro strap, and it measures levels of activity over the course of that week. So it gives you a proxy for when somebody's asleep and when they're awake. We asked people to complete a 7-day sleep diary, and we then did a 2-night sleep recording in the child's own home. And I've just realised that putting Stevie Williams on the slide, I still didn't mention him at the start. I moved him from the end because I tend to do that. So just to come back to that, sorry, Stevie Williams was my research assistant on this project. And it was him that went around the UK to the homes of families affected by SYNGAP1 to do the recordings in their own environment. The equipment you can see here, I don't know if you can see my pointer, but in the middle at the top, we have 2 participants who were wearing the kit. So it is all attached to the person. It all comes around into a sort of little box here that just sits just under your breastbone, and it measures all sorts of elements, which I will come to in a minute. It's just picking out bits that might be interesting for you from our results. We recruited 15 children with SYNGAP1 and 15 children without, who we call our control group. And in terms of the children's sleep habits questionnaire, we totted up all the scores and found that their score is worse sleep. And typically a score of over 41 indicates poor sleep. So you can see that the SYNGAP1 group had a score of 51.6 on average. You will also notice, if you look carefully, that the control group actually had an average of 42. So they alone could be described as having sleep problems. We don't necessarily understand why that might be, but it could be because having a sibling with SYNGAP1 might be disruptive in the household. And we do know that some of the participants co-slept with their sibling, because typically we tried to have somebody who was a sibling of the affected individual, so that when we were recording in the same house, it meant that there weren't so many environmental differences. So say a fire alarm went off, then it would have affected both of them and it wouldn't have skewed the results quite as much. So in terms of those sleep recordings, we measured EEG. So we've heard about this today already. So measuring the brain waves, the brain activity, and then to score different types of sleep, you need other measures. So there's measures of eye movement, oxygen levels, video, and the airflow coming and going as somebody breathes. We actually had to give up on that aspect because children nose there, but we were able to record pulse. Underneath, I'm not going to go into this in any detail just from a time point of view, but I thought I would just show you what's a sort of, we call this a hypnogram, what it looks like when you put a whole night's sleep recording together. So you may well be aware that there are different stages of sleep. And in this graph, if you're at the top level, then you're awake. And then as it goes down, right down at the bottom, we're in the deepest sleep. And in between, you have different types of sleep. The red lines are indicative of REM sleep, so rapid eye movement sleep. That colloquially tends to be referred to as dream sleep. It's not the only part of your sleep in which you dream, but dreams are particularly vivid during that period. So what did we find? Well, we discovered that although most of the sleep stages, so the sleep tends to be split into 2 main stages. You have non-REM sleep, and then you have the REM sleep that I've just described. The non-REM is split into 3 different sets of sleep. When we looked at those 4 different stages of sleep, the only one that was significantly different between the people with SYNGAP and our control group was the REM sleep. So there's something going on in specific aspects of sleep. What's really important about this, and comes back to what I was saying in terms of what we were hoping to achieve in this project, is that it is exactly the same finding as Dr. Gonzalez-Sulcer found in rat models of SYNGAP. Now that is important because if it means that tests are ongoing in the animals for potential therapies, then we can use the same technique that they use, so the EEG brain activity recording, in people, and therefore if we were to test the same medicines at some point, then we could directly compare across and see whether there is a response to treatment. So I've already mentioned this really, but just quickly, in terms of the rapid eye movement sleep, during this phase your brain activity increases, you get temporary muscle paralysis except your eyes and obviously your breathing. So this is why we look at eye movement as part of the sleep recordings. It's a very important sleep phase for memory, learning and creativity, and has the most vivid dreams. So clearly, if you're not having as much REM sleep, then this is going to impact on your daily function, potentially in terms of memory and learning, that sort of thing. So I just want to come briefly to the epilepsy side of things in SYNGAP, because it is frequently reported in some of the research papers. Everybody's diagnosed with epilepsy. In our situation, 8 of the 13 participants that we got successful sleep data on, so as I said before, we had 15 people with SYNGAP recruited. We got 13 people we had sufficient sleep data to analyze, and 8 of those in the SYNGAP group had a diagnosis of epilepsy. None of the control group had epilepsy. Out of those 8, only 3 had recordings on our sleep data that could be indicative of seizures. There was somebody else that we know from the sleep diary was having absent seizures during the day, but nothing was seen overnight. So we had 3 people with episodes that looked like they could be seizures. Again, this is important because of the rat data. So in the rats, as I've tried to show here on the left, there was evidence of absent seizures in quiet wakefulness. So when the rats were going to sleep or waking up. And hopefully you can see my pointer, because I'm just showing you here in the middle of the SYNGAP rat panel here, there's this change in all these brain waves that shows that the rat is having a seizure at that point. And then hopefully you can see on the right that in the top panel in particular, it's really quite similar. There's this point in the middle of the recording where it looks very much like a seizure. Some of our recordings, we did have confirmation later from the participants' neurologist that they thought it was seizures, or the parents reported back. But for others, it was our interpretation. Stevie, who did the analysis, is a very experienced sleep physiologist, but neither of us are neurologists. So we weren't able to offer the family any diagnostic information, but if we saw something unusual, we provided them with the data to share with their doctors. And just at the bottom here is another change in the recording that is indicative, we think, of seizures. So that's as far as we've got with our sleep research at the moment. We are still writing up the data and looking at various other aspects. But I hope that's been interesting. There are people I know on the call who, or potentially on the call, who may remember that I was actually in touch with the EEF 1A2 family group last year, because Kathy and I had talked about doing a study where we could compare sleep in EEF 1A2 in SYNGAP and also a third genetic condition. Unfortunately, we weren't successful in getting that funding, but I continue to look out for opportunities, and it may be that we can revisit that sort of idea at some point in the future. So thank you for having me.